Welcome, everyone. My name is Alexa Narowski, and I am a Genomics Education Fellow working at the Jackson Laboratory in Farmington, Connecticut, supporting the Teaching the Genome Generation program. Today, we'll be presenting two activities from our new curriculum modules on ancestry testing and on variation in the genome. During this webinar, we will discuss the new content that has been developed by our TtGG team and provide a live demonstration of two new activities from our variation in the genome and ancestry testing modules. This meeting will be webinar style. If you have any questions or comments, please add them to the chat as they arise. Our team will try to address questions in the chat as we go, and we will address any remaining comments and questions at the end of the presentation. This webinar is also being recorded and it will be posted on our website. We will move fairly quickly through the demonstration, but you will have access to the recording after the webinar if you want to revisit any of the content. First, I will begin by giving an overview of the Jackson Laboratory and our Teaching the Genome Generation program for any in attendance or viewing the recording who are not familiar with our program. So the Jackson Laboratory was founded in 1929 in Bar Harbor, Maine. We are a nonprofit biomedical research institution with a second research campus in Farmington, Connecticut. Our research focus is genetics and genomics, and we have a suite of education programs that aim to train the next biomedical workforce, as well as to instill knowledge of genetics and genomics in younger populations. One of those programs is the Teaching the Genome Generation program. And the goal of this program is to make genetics and genomics accessible to students and teachers by linking program content to personalized medicine and health, ethical decision-making, and everyday life. We also aim to celebrate the infinite variations within humans and lower the barriers for teachers to bring new content into their classrooms through detailed lessons and activities, tutor resources, and personalized support. Since 2015, our program has trained over 279 teachers, been implemented in over 180 schools, and introduced more than 21,500 students to modern genetics and genomics. We've had federal funding for this program from the National Institute of Health through their Science and Education Partnership Award since 2016, and we received a second phase of grant funding in 2021, focusing on new content development around math literacy and quantitative skills, and an expanded target audience to include pre-service teachers. In this webinar, we will present some of the new content we have developed as part of this initiative. Additionally, before I jump into the content, if you are attending or watching and are not yet part of our network, thanks for being here. And if this introduction has sparked your interest and you want updates from us on a regular basis, please consider signing up for our quarterly newsletter. The QR code and link to sign up are on this slide, and we will also share them again at the end of this presentation. So now I'll provide a brief overview of our new curriculum. Our TtGG team has been designing new lessons and activities in bioinformatics and genomics. The overall goal of this new curriculum is to teach math and data literacy. The lessons and activities will be hosted on our website and will be free and accessible to anyone who wants to use them. We are creating content in 4 different areas: cancer genetics, sequence comparison and identity, variation in the genome, and ancestry testing. Our cancer genetics and sequence comparison modules and their corresponding webinar recordings are already available on our website. And today you will hear more about our new variation in the genome and ancestry testing modules. First, I will present on our new variation in the genome module. I will start with an overview of the module and its learning outcomes, then provide some background information, and finally demo the first module activity that we have released. The variation in the genome module focuses on 3 enduring understandings: that genetic variations can impact sensory perception, which is a polygenic trait in humans, that polygenic traits are influenced by multiple genes and variants, as well as the environment, and that human genomic variation is something to celebrate. The module aims to help students address the question, how can we determine associations between genetic variants and complex traits, And through completing the activity, students will engage and develop a variety of skills, including data analysis and interpretation, computation, basic statistical analysis, pattern recognition, and constructing explanations. This is the table of contents from the Variation in the Genome module teacher guide, so you can see the organization of the module and its components. To start, there is the introductory content, which includes learning outcomes for the module and descriptions of all the lessons and activities. The next section includes all of the materials for each activity. I'll be giving a demonstration of portions of the activity later in the webinar, but for now I will provide a brief description. The first activity we have released for this module is titled Establishing Genotype-Phenotype Associations. There are two versions of this activity, a paper-based version and a spreadsheet version. The two versions follow the same general structure and cover the same biological and mathematical concepts. The primary difference is whether students perform calculations and document the results by hand or within a spreadsheet. You can either select one version for students to complete, or you can mix and match sections from each version. The demonstration in this webinar will primarily focus on the spreadsheet version, but I will point out differences between the two along the way. Both versions of the activity focus on how researchers establish associations between genetic variants and phenotypes or diseases. In this activity, students work through calculations and statistical analysis to establish an association between a specific genetic variant in the taster gene and bitter taste perception ability. Students are provided a real data set with the taster genotypes and bitter taste perception phenotypes from 60 individuals. In the first part of the activity, they count the number of alleles associated with each of the 3 bitter taste perception phenotypes, and then guided questions prompt them to identify any patterns in the data and consider what additional analysis might be useful. In the second part, students calculate allele frequencies based on their allele count data. And in the third and fourth part, students set up and perform a chi-square analysis. First, students calculate expected counts, which represent a random distribution of alleles across the phenotypes. And then students compare their actual counts to their calculated expected counts using a chi-square test to determine if there is a difference between their actual data and a random distribution of alleles. Finally, students draw conclusions based on their calculations. Through guided questions, students consider whether they have established an association or causation and how broadly they could generalize their results. So let's dive into the activity in a little bit more detail. As the students would if you were introducing or implementing this module, we'll start with an introduction to genome-wide association studies and bitter taste perception. The information I will cover here is provided as a short introductory reading at the beginning of both versions of the activity. You may be aware that many of our traits are influenced by both genetics and our environment. For example, an individual with a variant in the CDKN2A gene might have an increased risk of developing heart disease, but whether that individual develops heart disease is also dependent on other genetic variants and on their environment. Even someone who does not have that variant in CDKN2A can develop heart disease, especially if factors in their behavior such as smoking or environment such as exposure to air pollution lead to increased risk. For complex traits like our heart health, how do scientists identify associations between genetic variants and a trait of interest when there are so many other variables? One tool that scientists use is called a genome-wide association study, or GWAS. For a GWAS, scientists start by collecting genetic data from a large population with varied phenotypes for a particular trait or disease. Then the scientists look at millions of variants and determine if any of those variants are associated with a particular phenotype of that trait. Typically, science will look at single nucleotide polymorphisms, or SNPs. SNPs are a type of variant where just one nucleotide varies from person to person. For example, to try to find associations between genetic variants and heart disease, scientists would recruit a large group of people to participate in their study, some of whom have heart disease and others who do not. The scientists look at all of the participants' genetic variants and see if there are any variants that are more common in the group with heart disease than they are in the group without heart disease. For example, they may find one SNP that is present in a few people in both groups, another SNP that is present in many people in both groups, and a third SNP that seems to be much more common in the individuals with heart disease. The scientists would then perform statistical tests to determine whether any of the variants are more common in the heart disease group than would be expected just by chance. If they are, we would say that the SNP is associated with the trait or disease, as is shown for the third SNP at the bottom. In this activity, students will perform calculations similar to those involved in a GWAS to demonstrate an association between bitter taste perception and variants in the taster gene. The TASTE2 receptor member 38 gene, which is abbreviated TAS2R38 and which I refer to as taster, produces the taster protein, which functions as a receptor to perceive a wide range of bitter compounds. Bitter taste receptors, including taster, are proteins found on the taste cells of the tongue. There are 3 bitter taste perception phenotypes which students will investigate in this activity: non-taster, taster, and supertaster. Non-tasters do not perceive bitter taste. Supertasters perceive bitter taste and the taste is typically strong. And tasters perceive bitter taste, although the taste may be mild. While we group people into these 3 phenotypes, these phenotypes are subjective and correspond to a range of actual bitter taste sensing levels. Additionally, bitter taste perception is affected by multiple genes and by environmental factors, such as the foods we are exposed to and have regular access to. In this activity, students will look at variants in the taster gene. The taster variants are single nucleotide polymorphisms, or SNPs, which again means just that one nucleotide varies from person to person. There are 3 common SNPs in taster, which each occur at different locations in the gene. At the first taster SNP location, the nucleotide is usually either a cytosine or a guanine. At the second taster variant location, this nucleotide is usually either a cytosine or a thymine. And at the third taster variant location, the nucleotide is usually either an adenine or a guanine. There are different versions of this activity that focuses on each of the 3 SNPs, which I will discuss further later, but for this demonstration, I will focus on the first SNP, the C and G SNP. Now that you've had an introduction to some of the relevant concepts for this activity, I will dive into the activity demonstration. This activity uses a real genetic dataset with the aim of helping students understand the associations between complex traits and genetic variants. The dataset for the activity contains genetic data from a group of 60 individuals, including both their taster genotype and bitter taste perception phenotype. The samples were provided voluntarily, meaning that each person agreed to provide a sample. The samples were also anonymized, meaning that they were labeled with random sample names so that they cannot be traced back to the person who provided the sample. The data is provided to students in a table. For the spreadsheet version, it is provided in the spreadsheet, while for the paper-based version, it is printed in the handout. This screenshot shows just the first 5 rows of the table. In the first part of the activity, students count the number of alleles associated with each of the 3 bitter taste perception phenotypes: supertaster, taster, and non-taster, based on the data in that table. The purpose behind this step is threefold. First, the count data students summarize in this section is needed for analyses in the later parts of the activity. Second, summarizing the data will help students to identify patterns in the data. And third, this part of the activity starts a discussion about different ways of analyzing and representing data, which can be continued in part 2. Some skills that students will be building in this part include working with data tables and identifying patterns in the data. Guided questions at the end of this section reinforce this with prompts to identify any patterns in the data and consider what additional analysis might be useful. For the spreadsheet version specifically, some spreadsheet skills used in this part of the activity include sorting a data range and using built-in functions, specifically the COUNTIF function, or entering data into a spreadsheet. At the beginning of activity, students are introduced to this data set that they will be working with. But before students dive into their analysis, we provide some scaffolding to help them understand the data in the table, since students may have different levels of familiarity and confidence in working with data and tables. First, we provide an image of the raw data table with rows and columns numbered for reference. Then students answer several questions about the data in the table. Let's take a look at the table in the spreadsheet and talk through some of the questions. So if you decide to use the spreadsheet version of this activity, you and your students have the option to use either Google Sheets or Excel. I will be demonstrating using Google Sheets, but all of the functions I will use are either extremely similar or exactly the same in Excel. The spreadsheet is formatted with the raw data table on the left over here, and the analysis tables on the right over here. The paper-based version contains all of these same tables, just printed within the handout. When students open the spreadsheet or find the tables in their handout, they will start by looking at the raw data contained in Table 1. As mentioned, the worksheet contains questions to orient them to the data table, including what each of the columns represents, what a row represents, and how many alleles are included in the dataset. The goal of these questions is to make sure that before students begin their analysis, they understand that each row of this table represents one individual, that column 4 represents the bitter taste perception phenotype, and that column 2 and 3 represents genotype. There are 2 columns for genotype because everyone has 2 copies or alleles of each gene. For example, for this variant, individuals can have a C on both copies, as this person does, a G on both copies, as this person does, or 1 G and 1 C. Finally, we also want students to understand that empty cells, which are in gray, like these ones, represent missing data. For the spreadsheet version, the raw data table starts out sorted alphabetically by sample name, as you can see here. To organize the data and make it easier for students to analyze, the spreadsheet version of the activity asks students to sort the raw data table by phenotype to make it easier for them to count later on. This also introduces the spreadsheet skill of sorting a data range. So students will highlight the table either by clicking on a cell and dragging, by using Shift and their arrow keys, or by selecting one of the cells in the table and hitting Control-A on their keyboard to select the whole table. Then the student will click on Data, so go to Sort Range and Advanced Range Sorting options. If students have selected the column headers, such as sample name and phenotype, which I have here, they will need to select the checkbox labeled Data has a header row. Then students can select to sort by phenotype. The basics of this process work the same in Excel, although the menu labels and pop-up are slightly different. Instructions are provided in the activity for sorting in either software. Now we can see that all of the data has rearranged such that the samples are sorted into groups based on their bitter taste perception phenotypes. This will make it easier for students to count the number of alleles associated with each phenotype. In the paper-based version, the sorted spreadsheet is provided at the start of the activity since students will not be able to easily sort the data on their own. Next, as a first step in their analysis, students will count the number of alleles that are associated with each genotype. So the number of C alleles associated with each of the non-taster, supertype, taster, and taster phenotypes, as well as the number of G alleles associated with each. Table 2 over here is provided for students to record their results in. As with the raw data table, we provide scaffolded questions to ensure students understand the format of the results table before they begin their analysis. Students using the spreadsheet version have 2 options for counting and documenting the number of alleles. First, they could count manually. For example, if a student was to count the number of G alleles associated with non-taster, which is this section here, they would find that there are 30, and they could type that into the corresponding cell in Table 2. Likewise, you can pretty easily see that there are 0 C alleles associated with the non-taster phenotype, so a student could also type that into Table 2. The other option in the spreadsheet version is to use the COUNTIF function, which I will demonstrate by counting the C alleles for the supertaster phenotype. COUNTIF is a built-in spreadsheet function which will count the number of cells in a specified range on your sheet that meet a certain condition. So if the student would type equals and then COUNTIF and a parenthesis, into one of the cells of the table, and then select a range of cells that they want to count. In this case, we are looking at the supertaster phenotype. So we want to select the range that corresponds to the alleles from individuals who are supertasters, which is this range here. Next, the student would specify the condition. For this cell of the table, where we're in the C column, or C row, we are looking to count the C allele. So for our condition, we would type a comma after the range, and then the letter C in quotation marks. So this formula says count all of the cells in this range, which we know corresponds to the supertaster phenotype, if those cells contain the letter C. Now we can close the parentheses and press enter to see the result. There are 16 C alleles associated with supertasters. We could confirm this by checking the supertaster section, which if we were to count, indeed does have 16 C alleles. So with the spreadsheet version of this activity, you can allow students to select one of these options for counting alleles, ask them to do one particular option, or you can have them do both as a way to check their work. For the paper-based version, students only have the option to count the data themselves and write the results into the table in their handout. If we were to complete the counting for all of the different alleles and phenotypes, we would see these results in table 2 over here. At this point, we can see in both the sorted raw data table and the table 2 allele counts that there's a pretty clear pattern in how the alleles are distributed across the different phenotypes. So after doing the counts, students are prompted to reflect on the data they collected and look specifically at patterns. So there are several reflection questions for this part of the activity. The first reflection question guides students to think through what patterns they noticed in the data. So what patterns might the students notice? Looking at the results again, we can again see a pretty clear pattern in the distribution of alleles across the non-taster and supertaster phenotypes. Students may also notice that the taster phenotype, which is in between the non-taster and supertaster phenotype in terms of bitter taste perception ability, has a more even split of alleles, although it's not quite 50-50. So after students spend some time identifying these patterns, additional reflection questions prompt them to think through whether the data suggests that there could be an association between this variant and bitter taste perception phenotype, and what additional data collection, analysis, or representation would help them to better support that hypothesis. Connecting back to the skills students are using in this activity, these reflection questions aim to get students to identify patterns in a data set, interpret those patterns, and use the patterns to suggest additional analysis. In the next part of the activity, students will calculate allele frequency as another way of analyzing and representing this data. So in the second part of the activity, the student's primary task is to calculate the allele frequency of each allele for each phenotype. For anyone who's not familiar with allele frequency, I will talk more about it in a minute. The purpose behind this step is again threefold. First, it introduces allele frequency, which is a common measure in genomics research. Second, the allele frequency values calculated in this part are needed for later parts of the activity. And third, since the later parts of the activity introduce statistics, which may not be suitable for all students, this provides a simpler calculation for students to do to help them to get comfortable doing math and biology classes. Some skills that students will be building in this part of the activity include working with data tables, identifying patterns in data, and performing computations. For the spreadsheet version specifically, some spreadsheet skills used in this part of the activity include having students create their own simple formula to perform the allele frequency calculations, or entering values they calculated manually into the spreadsheet. So in the second part of the activity, students will use the allele count data that they compiled in part 1 to calculate allele frequencies. Allele frequency refers to the number of copies of an allele, in this case GRC, across all of the alleles in the population, or in other words, the proportion of each allele observed in the population. The equation we will use for allele frequency is nu over 2n, where the variable nu in the numerator is the number of each allele in the data set, and the variable n in the denominator is the total number of diploid individuals in the dataset. In part 1, students counted the number of each allele in the dataset, so their allele counts table from part 1 will contain the values for new. Additionally, since n represents the total number of individuals, and each individual has 2 alleles, 2n is equal to the total allele count. So we can also write the allele frequency equation as the allele count of a specific allele in the population divided by the total allele count in the population. For example, students would use the allele counts in Table 2 to calculate allele frequency. To calculate the allele frequency of the G allele in the populations of individuals with the taster phenotype, for example, they would use the allele counts table to find that in our population of 60 individuals, we have 30 tasters with known alleles, corresponding to 60 alleles total. Of those 60 alleles, 35 are G alleles. The student could then calculate the allele frequency of the G allele in the taster population by dividing 35 by 60, which gives 0.583. After students are introduced to the concept of allele frequency and perform an example calculation, as we just did, they are introduced to the next results table where they will record the results of their allele frequency calculations. As before, we provide an image of the table with rows and columns numbered for reference. Then students answer several questions about the table layout. The questions are less thorough than they were for the first table, so that students don't feel the questions are repetitive, and so that the scaffolding is slowly reduced over the course of the activity. In this part, students are asked to identify where they would enter the results for a particular calculation. For example, one question asks them, into which cell would you enter the data for the frequency of G alleles in the total population? And students should put an X into this cell here, which is in the G row and the total population column. Again, these questions aim to make sure students who are not used to working with tables take some time to figure out how they should enter the data before they start performing calculations. In other words, helping students build that target skill of working with data tables. Now I will switch back to the spreadsheet so you can see what the process and results for this part of the activity will look like. So after students get familiar with the table, they can perform calculations and enter data. As with the first part, the primary difference between the spreadsheet version and paper-based version is whether students are typing data into the spreadsheet or writing it down on the table in their handout. Students will already know some of the values from the example calculations in the handout and can enter that data directly. For example, we already calculated the frequency of the G allele in the taster population to be 0.583. So we can enter that into the allele frequency table, which is here. Oops, I already have the answer in there. Specifically in the G row and the taster column, which is the cell right here. Students with either the spreadsheet or the paper version can continue calculating in this way, performing the calculations by hand and then entering or writing down the data. For the spreadsheet version, this option builds the skill of data entry. Students working on the spreadsheet version also have the option to create their own spreadsheet formulas to calculate the frequencies. For example, to calculate the frequency of the G allele in the non-taster population, which would go into this cell here, students should know that the value of the cell should be equal to the allele count of the G allele in the non-taster population, which is in the Table 2 allele counts data here, divided by the total allele count in the non-taster population, which is here. So in the proper cell in the allele frequency table, the student would type equals, then select the non-taster G allele count, then type a slash to represent division, and select the total non-taster allele count. And then press enter, and it will return the calculated allele frequency. Again, for the spreadsheet version, you can ask students to pick between manual calculation and data entry, or spreadsheet formulas. You can choose the method for them, or you can ask them to use both. Your choice may depend on whether you prefer them to get more practice performing the calculations manually, or more practice using the different functionalities of spreadsheets. Once students complete the full table, they should obtain these results. As in part 1, students should hopefully notice that there's a pretty clear pattern in how the alleles are distributed across the different phenotypes, specifically for the super taster and non-taster having a specific association, and with taster being almost 50-50, but not quite. At the end of this part, students are again prompted to reflect on the data that they analyzed. And I'll switch back to the slides to show you those questions. So the first reflection question for part 2 again guides students to identify any patterns in the data. The second reflection question reminds students that the data in Table 2 and Table 3 come from the same raw data, but show different ways of analyzing and representing that data, and then ask them to consider which set of data is easier for them to understand. Connecting back to the skills students are using in this activity, these questions aim to get students to identify patterns in a data set, interpret those patterns, and discuss different ways of analyzing and representing data. So at this point in the activity, students have analyzed the raw data to determine the distribution of the different taster SNP alleles across the different bitter tasting phenotypes. However, we still have not answered our primary question. Is there an association between this genetic variant and bitter taste perception phenotype? To thoroughly answer this question, we need to perform some basic statistical analysis. So in parts 3 and 4 of the activity, students will set up and perform a statistical test called a Pearson's chi-square test of independence. You may be familiar with chi-square tests in other contexts, such as Hardy-Weinberg. This activity uses the same test and concepts, just with a different application. In this activity, the chi-square test is used to evaluate whether observations of measures on two variables, in this case allele and phenotype, are independent of each other by comparing actual counts to expected counts. Like with Hardy-Weinberg, we compare our actual counts to our expected counts to see if there's a deviation from the expected distribution of alleles in this population. In this activity, the actual counts are the allele count data from the first part of the activity. However, students still need to calculate expected counts. So the primary student task in part 3 of the activity is to calculate the expected count of each allele for each phenotype. The purpose behind this step is twofold. First, the expected counts are necessary for chi-square analysis. And second, these calculations provide additional practice of data analysis and computation using a different equation and calculation type than allele frequency. Some skills that students will be building in this part include working with data tables, identifying patterns in data, performing computations, and also the start of some interpretation of their results. For the spreadsheet version specifically, students again have the opportunity to practice creating their own simple formula to perform the expected count calculations, or entering values they calculated manually into the spreadsheet. So even if you are familiar with chi-square tests in the context of Hardy-Weinberg or another context, the method for calculating expected counts for this application might be slightly different. The handout explains the reasoning behind the expected counts equation and provides calculations, practice calculations before asking students to complete the analysis. In this application of a chi-square test, the expected counts represent what the allele counts would be if there was no association between the variant and phenotype. In other words, the expected counts represent a random distribution of alleles across the phenotypes, such that each phenotype has the same allele frequencies as the total population. If we focus on that second part, we can think about this in terms of allele frequency, which students already calculated in the previous part of the activity. So expected count calculations in this context represent what the allele counts would be if all of the allele frequencies for all of the phenotypes, which are highlighted in green, match the allele frequency for our total population, which is highlighted in blue. So if we were to calculate an expected allele frequency based on our expected counts, the allele frequency table would look like this, where the allele frequencies for each phenotype matches those for the total population. Now we would just need to figure out what allele counts would create this allele frequency table. Since we calculated allele frequency earlier, we can use the allele frequency equation to set up our expected counts equation. The expected allele frequency for a specific allele will be equal to the expected count for that allele and phenotype divided by the total allele count for that specific phenotype. As we just discussed, because of how expected counts are defined in this case, we know that the expected allele frequency is equal to the allele frequency in the total population. So we can substitute those values into our equation. If we rearrange this equation, we could calculate the expected counts. So students will calculate the expected count of a specific allele for a specific phenotype by multiplying the allele frequency of that specific allele in the total population by the total allele count for that specific population, for that specific phenotype. The allele frequency values should come from the total population column of the allele frequencies table, and the total allele count values should come from the total row of the allele counts table. The student worksheet has guided questions to help students understand the concept of expected counts, identify the cells in these two tables that contain the data relevant to these calculations, and practice the calculations. Once students have had the opportunity to practice a few calculations, they are introduced to the results table for this part of the activity. Then students perform the remaining expected count calculations and enter the data into the results table. As with the previous parts of the activity, students using the paper-based version will calculate all of the values by hand and write them into the table in the handout, while students using the spreadsheet version can choose to calculate by hand or use spreadsheet formulas. Students will obtain the following results, and at the end of this part, students are again prompted to reflect on any patterns they noticed in the data. The reflection questions also ask students to compare the expected counts to the actual allele counts from the earlier part and determine if they can draw any conclusions from this comparison. In this comparison, students should hopefully notice that the expected counts and actual allele counts do not match, and depending on their level of familiarity with statistics, and also how much they were paying attention to the introduction reading at the beginning of this part of the activity, they might suggest that they should perform a statistical test to confirm whether the difference they observe is real or is likely just due to chance. And in the next part of the activity, that's exactly what they'll do. So in part 4 of the activity, students are tasked with performing and interpreting the results of a chi-square test. The primary purpose behind this step is that it completes the statistical analysis and provides a basis for students to draw conclusions about the data set. Some skills that students will be building in this part of the activity include data analysis and interpretation, basic statistical analysis, and constructing explanations. For the spreadsheet version specifically, students again have the opportunity to practice using built-in functions, specifically the function for chi-square tests. In this part of the activity, students will perform a Pearson's chi-square test of independence using their actual and expected allele counts that they calculated previously. As a reminder, this test evaluates whether observations of measures on two variables, in our case allele and phenotype, are independent of each other. At the beginning of this part of the activity, students are introduced to the chi-square test and the null and alternative hypotheses for this test. A null hypothesis represents the assumption that there is no difference between groups or no relationship between variables. For this test, our null hypothesis is that the count of each allele is independent of phenotype, or in other words, that the count of each allele is the same or very similar for each phenotype. The alternative hypothesis is our test or the claim that we are trying to investigate. In this case, our alternative hypothesis is that the count of each allele is dependent on phenotype, or in other words, the count of each allele is different for each phenotype. Students are also reminded that for statistical tests, it is important that we establish a level of significance before we perform the testing. The level of significance, also called the alpha value, represents the maximum probability you are willing to allow your results to be due to chance. Your chosen alpha value can change based on your research question and on how much potential error you are willing to tolerate in your results. In this case, we will choose an alpha value of 0.05, representing a 5% probability that our results are due to chance. So if the outcome of our test suggests that the probability that our results are due to chance is less than 5%, we can reject our null hypothesis and conclude that there is an association between this taster variant and bitter taste perception phenotype. So after being introduced to the test and hypotheses, students will perform the chi-square test and evaluate the results. In the spreadsheet version, students use the built-in chi-square test function to compare their expected and actual count tables. This function returns the p-value for the chi-square test. The p-value, or probability value, represents the probability that any differences observed are between the expected and actual counts are due to chance. For the paper-based version, the p-value is directly provided to the students. In either case, students will obtain a p-value of 2.16 times 10 to the negative 10. At the end of this part, reflection questions guide students through interpretation of their results. First, students are asked to determine whether the p-value meets the established criteria for statistical significance. Then, students are asked to interpret the outcome in the context of association of this taster variant with bitter taste perception phenotype. And finally, students are asked to reflect on whether this outcome reflects their earlier conclusions about the relationship between variant and phenotype. At the end of the activity, there is an additional reflection section that provides an opportunity for students to think about the implications of the results and the extent that their conclusions can apply to a more general population. The questions include, what can we conclude about allele phenotype association from this data set? And how broadly can we generalize our results given what we know and don't know about our sample population? These questions intentionally do not reference the statistical analysis, so that you can still use the reflection questions with your students, even if you have them skip the statistical analysis portion of the activity and stop after calculating allele frequencies. These questions aim to get students to think about both their conclusions and the implications of those conclusions. Additionally, you may recall that at the start of the activity, we actually introduced 3 SNPs in taster. Each taster allele that an individual has will have one specific nucleotide at each variant location. This combination of the 3 variants on a specific allele is called a haplotype. For example, one possible haplotype is GTA, representing a G at the location of SNP1, a T at the location of SNP2, and an A at the location of SNP3. Although I have only analyzed the data for one of the SNPs in this demonstration, we have worksheets available for each of the 3 SNPs. This means that you could have students perform calculations for all 3 SNPs, or you could have students complete this activity as a jigsaw, where small groups each work on different SNPs and then get rearranged into new groups where they have to present to their peers on the results for their assigned SNP. In either case, or even if you only have students work through one of the SNPs, some of the reflection questions ask students to reflect on what conclusions they can draw about the combination of all 3 SNPs. These questions include, from this data set, do we know which combination of variants were inherited together on a single chromosome in a single individual? And can we draw any conclusions about haplotype-phenotype association from this data set? Finally, the reflection questions also connect back to the concept of genome-wide association studies, or GWAS. Students are asked to reflect on whether they can conclude that the variants cause an individual's bitter taste perception phenotype. We specifically ask them to only take their own results into consideration because there is a known mechanism that connects taster variants to bitter taste perception. However, for the purpose of this exercise, we want them to acknowledge that they can only claim an association, not causation, based on their analysis. This question allows us to connect back to GWAS, since a GWAS can only establish correlations or association and cannot be used to establish causation. We ask students to reflect on why it might be important to establish a correlation between variants and a phenotype, even if they cannot prove that those variants cause the phenotype. And finally, to connect back to the broader idea of bioinformatics, we ask students to reflect on this experience of performing calculations for one variant and consider why computational tools and automation are important for GWAS studies, which evaluate millions of variants across much larger populations. Reflecting back on the overarching goals of this module, this activity provided an opportunity for students to determine an association between a genetic variant and a complex trait using a wide variety of data analysis and math skills. While this is the only activity we have released for this module as of now, we plan to develop additional activities that focus more on how traits can be influenced by multiple genes in the environment. And that concludes the demonstration of the Establishing Genotype-Phenotype Association activity. I will now pass it over to Christina, who is going to discuss the new ancestry testing module.
Okay, I'm pleased to kick off the next section on the next module that we have ready for you, and that is the ancestry testing module, where we'll focus on sequence comparison. So for this portion, I'd like to give an overview of the entire workshop, and we'll go over the ancestry testing module as a whole, what it's comprised of, and then we'll dive into one of the activities, the sequence comparison activities, and go through the various parts from the scenario background to some of the calculations and work that the students will do, and conclude by providing some additional information and resources associated with the lesson. So let's get started. Let's talk about this module as a whole. So this module aims to introduce students to the concept of ancestry testing using a narrative to frame the learning activities. Students will follow the fictional story of Sam, a character in high school who wants an at-home ancestry DNA test for their 18th birthday. Each activity will begin with a scene from Sam's life. So students will meet Sam and learn that their family has kind of mixed feelings about the ancestry tests. Sam then approaches their biology teacher at school to learn about really how ancestry tests work. Sam will learn how genetic tests inform about more than just ancestry. And we leave Sam kind of reconsidering what this ancestry test means for them and whether or not they should take it. So as a whole, the module presents several essential questions for the students, and this includes how does ancestry testing work? Why do ancestry test results change? If you've ever taken an ancestry test yourself, you may have received several notifications that over time your results change. What do ancestry test results mean for me, for my health, and for my family? How does my ancestral DNA information connect with my identity? And then during understandings that we're hoping to build in this lesson, in these lessons include that DNA is inherited across generations, but also is lost. Not all of our genealogical ancestors, those in our family trees, are genetic ancestors that are contributing to the DNA that we have. Ancestry testing typically involves sequencing of single nucleotide variants. It's not whole genome sequencing. Ancestry reference genomes are aggregate single nucleotide variant data from various numbers of individuals. And ancestry information can inform about more than just ancestry, in addition to personal and family history, as well as health outcomes. So the skills built throughout these lessons include data table usage and interpretation, computation math skills, asking questions, formulating hypotheses, problem solving, pattern recognition, and constructing explanations. And this webinar will focus on just a few of these in this next activity that I'll be going through with you, and that's the sequence comparison activity. So let's jump into the scenario and the background that the students will be presented with first. So like I mentioned, each activity centers around, is framed with a narrative. So in this activity, we are presented with Sam approaching their biology teacher, learning how ancestry tests are performed. They're very excited about this ancestry test and think their whole genome is going to be sequenced, but in reality, their teacher says, actually, companies really only look at a fraction of the genome when they do these types of tests. So the goals of this activity, students use spreadsheets, electronic spreadsheets, to sort SNP data, genotyping data from an ancestry testing company, a fictional company, to perform allele frequency calculations and interpret the results. So background is presented first to the students, and we start off with genetics and inheritance. So just as an overview, we'll go through this as well for this webinar. Humans are diploid. They have diploid genomes, which means that we have 2 copies of every piece of DNA. 1 copy is passed from each gamete, 1 copy from the haploid sperm, and 1 copy from the haploid egg. This is what we see in this example here, and these sperm and egg cells come together to create the offspring, as is shown here. So we know that human genomes through genetic studies are 99.9% identical between individuals. only 1.1% of our DNA is different between individuals. And it's this variation in the 0.1% of DNA that are the genotypic differences that lead to the phenotypic differences that we see around us. So when only one DNA nucleotide varies, this is called a single nucleotide polymorphism, or a SNP, as we heard about earlier today in this webinar. So for example, that's illustrated here, we're at the same DNA locus. In the sperm cell, this cell carries a G allele, whereas in the egg cell, this carries an A allele. So this means that the offspring will inherit both the G and the A, and they will be heterozygous at this location. And because humans have two alleles at each genomic locus, that means that they can be either homozygous or heterozygous for at any locus. So the genotype of the offspring, of course, depends on the DNA that they inherit from both biological parents. So that's what we're seeing here in this illustration. And each site of our genome can be examined in the same way to observe which individuals carry which DNA variants. So if we look at a different locus, a different single nucleotide polymorphism at a different site in the genome, we observe here that the sperm cell carries an A allele, the egg cell carried a T allele, and the offspring now is a heterozygote for both of those alleles that were inherited. We also introduced the students to the concept of allele frequencies in this module. So similar to what was shown previously in this webinar, we know that counting allele frequencies in individuals, counting alleles in individuals reveals allele frequencies. And this is a measure of the distribution of all the DNA variations or all of the alleles in a population. And it can be calculated very simply once you know the number of each allele observed in your population, as we saw earlier in the webinar. And so we present students with this, calculation, and we share that for the allele frequency, every allele must be calculated. So there's a separate calculation for each allele for the C's, for the T's, the A's, and the G's. So let's go through an example. We provide this in the activity. We return to our example of the kind of blue and yellow DNA in the offspring that inherited. Parent 1 now has parent 1 and 2 have the full genotypes revealed, as well as the genotypes of the offspring. So if we count how many of each variant allele we detect in this particular data set with 3 individuals, recall at every DNA locus, there are 4 possible variant alleles for the 4 DNA nucleotides. So we can make a very simple table and perform our counts and record them in this table. So at this DNA site that we're looking at, we can count how many C, T, A, and G alleles we find amongst these 3 individuals. And we can record that here. Of course, we see no C or T alleles present. And we find that we have 3 A alleles total and 3 G alleles total. And we know that our data set are these 3 individuals, which means we have 6 total alleles. We can kind of count them manually from the tree, the pedigree here, or we can count them from this table that we just calculated. We also know that there are 3 diploid individuals, each with 2 copies of DNA. Now, to perform the allele frequency calculation, we basically just plug in the numbers that we just calculated. Again, each allele frequency is calculated for every allele. And when we plug in the numbers that we calculated up above from the counts, we find allele frequencies of each of the alleles in this particular population. Recall that allele frequencies represent the fraction of each DNA variant observed in the population, but what is that population? So in this module, we also emphasize that the population matters. We know that in this population, we had 3 individuals and they were biologically related, which means that they share DNA. So allele frequency data that we get at the end of these calculations can really change based on the population data set that we're working with and the diversity of individuals. So let's consider again the data that we had before. So the allele frequencies are shown here now as percentages in this pie chart. And at that same DNA site, in the 3 individuals, if we expanded that to, let's say, 50 individuals in another data set, we might find that those allele frequencies are drastically different because we had a larger data set now with more individuals with more diversity between them. Similarly, if we looked at another DNA site, we would expect that these numbers are different for the different alleles because of the genetic variation that exists and that every DNA site is not the same as the next. We also introduced the students a bit to the concept of sequencing and share that ancestry testing companies typically perform a type of SNP genotyping sequencing at DNA sites with known and high variation. So a wonderful video from the Personal Genetics Education Project is included in this part of the lesson so that students can learn how ancestry testing works. This can be watched in class as it is quite short or assigned as pre-work. Interestingly, some medical tests also use similar technology of this type of SNP detection to detect known variants. And also whole genome sequencing is not typically performed in ancestry tests, which may be a misconception that your students have. So next, we'll dive into the meat of the activity where we do some calculations on our spreadsheets. So a reminder that the scenario was the student Sam approached their biology teacher wanting to know how ancestry tests work because they had this misconception that it was a whole genome sequence. And Dr. V, the teacher, wanted to clarify that this was actually a SNP genotyping situation. And so Dr. V teaches the class how ancestry tests work. And so our students in this lesson are kind of following along with Sam and their classmates. So the example here is of an ancestry testing company called 23 Chromosomes that's building a reference DNA database. 23 Chromosomes selected 50 individuals from 5 regions around the world who volunteered to donate their genetic information for the company, and they'll use the genetic information from these individuals to build their own reference database. These individuals are represented from 5 global populations, labeled African, Asian, European, Native American, and Pacific Islander. So throughout this lesson, the students' tasks are to observe, sort, and then analyze the data to calculate allele frequencies at each DNA site for each population, just as an ancestry testing company would when making their reference databases. So the students perform data analyses in spreadsheets, digital spreadsheets. In this case, we're using Google Sheets. And throughout the activity, we recommend a jigsaw approach, but any approach is also fine. So in this activity, 2 options for spreadsheets of the genotyping data from the ancestry testing company is provided. One master spreadsheet can be provided where individuals are listed in one column from the different geographic locations. And then similarly, in the next subsequent columns are the genotype data for the various 5 DNA sites that are examined. This is nice to have one data set, one table to look at, but perhaps for some students that aren't as familiar with tables and sorting through the data, this might be a little overwhelming and it's a very big data set to work with. So another option we have is separating this out into 5 individual spreadsheets, each with just one table, looking at one SNP at a time. So the same individuals with the genotype data of the one DNA locus, and then 5 of these spreadsheets for each of the DNA loci. So in this activity, we recommend that either students individually as a class, we guide them through observing the data just to get familiar with the tables in the spreadsheet, the data that they'll be working with. And then in small groups, they work to organize the data, to count the number of alleles, calculate allele frequencies, and then coming back as a class and comparing their information that they gathered across the populations. So we'll do this together. So first, we instruct the students to just observe the data. We get a snippet of one of the data tables here. So again, this is data from the fictional ancestry testing company 23 Chromosomes with the individuals reported here and the genotype data for each DNA site reported in the next columns. And similarly to the new variant module that Alexa debuted, we walk them through reading the table with headers, columns, rows, etc. So they learn to identify spreadsheet features, as well as learn about the actual data that's coming through in this company. So how the individuals are coded with their country abbreviation, as well as their patient identifier or their individual identifier number, the 5 populations that are studied here, as well as the different genotype data that is coming through. So next, they're asked to organize the data. You can see that it's all kind of jumbled up based on just individuals in any random order. So the students are asked to work in their assigned spreadsheet to sort based on population to make the subsequent calculations easier to do. And so through a series of guided steps, the students are instructed how to select the desired data, use functions within the spreadsheet to sort the specific range in order to get a sorted table. And now you can see these are in alphabetical order, numerical order, sorted by, now we see first African individuals, then Asian individuals, and then their corresponding genotypes here. The next part instructs students to count alleles. So they work with the data that they just sorted in the first spreadsheet in order to count the alleles in each population. So again, through a series of steps that I won't go through in detail in this webinar, but just kind of outlining for you, the students are instructed to approach each population individually now that they're sorted. And for example, highlight the African population and use the next data table that's provided in order to count the alleles that they observe. There are 10 individuals, so 20 total alleles. This can be done manually or through spreadsheet functions. And the students are expected to do this for all of the 5 populations at this DNA site. Finally, as this information builds on itself, the students get to calculate the allele frequencies observed in the populations that they're looking at. So they use the counts data that they just calculated to now calculate the allele frequency. And just as in our example earlier, once we have that information, it's a really simple plug and chug. We can plug in the number of the count that we just generated to the total allele count that we know from the individuals, the total number of individuals in our population. And so the students can enter that information into their equations and receive the final allele frequency calculations at the end in the table and fill this out for all of the individuals at all the populations at each DNA site. So now they have the actual data that ancestry testing companies do to look at the distribution of alleles across populations. So the students are asked to come back and compare allele frequencies with their peers, especially if they were in the jigsaw groups, to kind of come together and report out the results that each group found individually. So for example, there are 2 tables here. This one is the allele frequency data at one of the DNA sites, and this one is at a second DNA site. The students are asked to compare within a table at one DNA site. Why are different populations different for the allele frequencies? And also across DNA sites, why might these be different? So there's a lot of pattern recognition, inferences, making hypotheses, etc. The activity ends with 2 parts on ethics and careers. The activity on bioethics asks students to really think about where reference population data comes from. What is the origin of this information? And so there's a very simple prompt here where a map is presented and students are asked to circle and label the areas that correspond to each population used by 23 chromosomes. So if you remember, there are 5 populations. And so the students are asked to label where in the world these 5 populations come from as best as they can. And so we expect that students would have very different answers. For example, there might be one student, if you're US based, you might think that Native Americans, really are only thinking about the United States. So for example, the Native American population, they may only consider the continental United States. Or Pacific Islanders, they may restrict only to the very small island nations that they know of in the Pacific. Whereas another student might identify that Native Americans actually comprise all of the Americas, and also Pacific Islanders comprise all of the island nations that they know of below the Asian continent. So you may see a lot of different answers in your classroom, and that's what we expect. And we ask students to think about, which version is correct, compare with your colleagues, which regions aren't highlighted, what countries might span different continents that you know of, and how might that influence what country is defined in each region. Thinking also about size, how many countries are in each region, how large is each region, and really history. Getting them to think about how the social and historical boundaries that we place in the world can influence something like these genetic results. Finally, we end with 2 career exploration tasks for the students. Students perform self-directed exploration of jobs and careers that correspond to this lesson. So earlier we learned about the information coming from the Ancestry testing company, 23 chromosomes, and certain jobs are highlighted like bioinformaticians and genetic bioinformaticians that may process some of this data, as well as genomic technologists that might actually run the sequencing tests in the laboratory. And so students are asked to pick 2 of these careers at least and explore data on known sites, job sites, such as the degree and training that's needed, the salary estimate, a description of the work, etc. And finally, students are asked to also reflect on these jobs after meeting some professionals, at least meeting them as much as they can in a virtual environment. We at JAX have a few career exploration resources, including our Career Chats virtual series, as well as some write-ups of people and jobs. We also point to some external sources of really great profiles of scientists across the world in different types of jobs. And so we ask students to find at least one of these profiles and reflect on it. Really describe the work that they're doing, ask what traits match their skills and interests, and inquire what they might want to learn more about. So hopefully making this a more real life connection to them and showing them that there are real people out there and real jobs that they could explore themselves. So to wrap up the ancestry testing module, I just wanted to share some more information. That at the beginning, I did present that the module overview has a lot of questions and understandings that we did not talk about. So there is definitely more to come. Some of the content that additional topics that we're interested in presenting in, for example, this concept of lost DNA. And so I'll just go over this very briefly. We know that every individual, when their gametes are being formed, go through the process of meiosis and recombination. And the process of independent assortment means that all the DNA basically gets shuffled around randomly. And so any part of your DNA may get recombined and passed on to the next generation. So if we just took a sampling of this individual's possible gametes, which are shown here, just some of the random samples that are possible of their gametes, we see that an offspring that might inherit some of these, none of them will actually inherit the G allele that the parent has here at that first DNA site, and only one individual will inherit the T allele at the second DNA site, just by chance. So this is some of the lost DNA that might be present or absent in families, and that's an exploration, a topic that we're going to explore in some of the other content. And just a reminder that any genetic test tells us about our DNA, right? And we know that DNA is inherited, which can tell us about ancestry, but because DNA is shared in biological families, it also tells us about our family and who might not be in our family, who might be in our family that we didn't know about as well. So sometimes these ancestry test results have big implications for folks across the world as they learn different information about not only themselves, but their relatives. Also, DNA, of course, is our body's instruction manual, and sometimes the results that we get from any sort of DNA study, even ancestry, could be linked to some health outcomes as well. And so it's important to understand the full implications of any sort of genetic test, even just an innocuous ancestry test. So as I mentioned, this module was the sequence comparison and ancestry testing module. We have a teacher guide coming soon. It's not quite out yet. But in addition, there are 3 other modules that are being planned to explore some of these topics that we talked about, really diving into reference populations and as well as implications of ancestry and human health, as well as ancestry and personal identity. So please stay tuned for those parts. And finally, I just wanted to share that this lesson was intentionally designed to be linked with another lesson from the Personal Genetics Education Project. They have really a fantastic lesson called How Does Ancestry Testing Work? And our lesson is focused more on how ancestry tests are performed. So students get to perform that SNP genotyping themselves, and they learn how to calculate the allele frequencies, where those numbers come from. The PG Ed lesson is kind of flipped from this perspective. And so if your students did that lesson, they would focus on how ancestry tests are interpreted. So students get to estimate an individual's ancestry results based on allele frequencies. So this is also a fantastic lesson. You could do both and kind of link and be able to share, or one or the other. So to conclude the entire webinar, I will end with just an overview of some of the teacher resources that we have at the Teaching the Genome Generation program, especially for those that might be tuning in that are not familiar with our resources and our program. As Alexa said at the top of the meeting, we are really pleased to open up our newsletter to more than those that we have trained in person. So if you are interested in signing up to our quarterly newsletter, please feel free to scan this QR code. We do share program updates as well as some other fun facts about genetics in the news, some science education resources, and we often do different features such as career corner spotlights and the like. A reminder that we do have virtual professional development opportunities as well as in-person training if you are in New England. We have a genetics learning resources website where all of the lessons that we presented today, as well as pretty much everything else that we have in our suite of resources, is freely available for you to download and access online. And we have a lot more than just the TtGG program that we've done. And we would encourage you to check out all the different things that we have there that all correspond to learning opportunities in genetics. And finally, we'll share that we have an ongoing collaboration with Lab Exchange, the virtual classroom, and we have all of our content and more, including this gel electrophoresis simulation that is free and available on Lab Exchange. If you're already on that platform, you can remix our content in with your current pathways. And if you're not on Lab Exchange yet, it's really a fantastic educator resource. So we do encourage you to check them out. And with that, I will conclude this webinar. Thank you so much for coming. To those that attended, thank you for watching if you're watching afterwards. If you're live, we're very happy to take questions now. Please use the chat and we would be happy to respond. And if you would like to contact us at any time, we really welcome your emails. Check us out online and you can follow us on social media as well. Thank you very much.